Good afternoon. If I could please ask everybody to take their seats. Thank you. And welcome. Welcome to a very important event. This is the 40th anniversary of the Tyler Prize. I am Donald Manahan. I am a professor of biological sciences and a dean of students at the University of Southern California. And on behalf of the executive committee, which I would like to immediately acknowledge, if the executive committee could please stand, the Tyler Executive Committee. Thank you. Come on, Ralph, up you get. There you go. Ralph and I go back many, many years. I won't introduce them all individually. I will, however, just mention that Owen Lind at the back of the room, who's the chair of the committee. We'll be introducing them in more detail later this evening. So I'd like to thank the Tyler Prize Executive Committee on behalf of the university. What the role of the university in this is really just to help the Tyler Prize Executive Committee to make these great decisions in choosing our laureates each year. Of course, the university also plays an administrative role. I see Amber at the back of the room, who is the administrator of the Tyler Prize. There you go, Amber, waving to everybody. Thank you, Amber. She's taking pictures. But the main reason we're gathered is to honor the laureate for 2013, Diana Wall. Diana, this means a lot to me personally. I've known Diana for 25 years. We've worked in Antarctica together over the years. So this is important professionally and personally. But let me just start with the professional side by way of introduction to her lecture this evening. She is, as I said, the 213, the Tyler Prize winner for environmental achievement. And it's awarded to her for her outstanding research in environmental work, and I'll talk a little bit about more of that in her introduction in a moment. Professionally, Diana is a university distinguished professor at Colorado State University, and among her many other positions she holds at an institution, she's the director of the School of Global Environmental Sustainability. There's no question that Diana has an extraordinary career. Her mom is here, so Mom, you take notes on this one in case you missed anything. Mom, would you stand up, please, and be acknowledged by the crowd? Come on, come on. Wow, thank you. So what has Diana done? Well, what hasn't she done? She, for example, in terms of, look at the front here, about serving science and society, the motto of AAAS. Diana really has lived that life. She has been president of 6 major scientific societies, 6, including the very prestigious Ecological Society of America. She is appropriately elected in this building, the building for the American Association for the Advancement of Science. She was elected a fellow of AAAS, and among her many other awards, she has a feature, as we call it, in Antarctica named after. Wall Valley in Antarctica is named after Diana for her distinguished research in the dry valleys. She has published over 200 papers. And in a word, what she's worked on is the whole question of the structure and function of soil ecosystems. Now, why we care about soil and environmental sustainability is pretty obvious anytime you eat anything. Soil and food. What you may not know is the 7 billion plus people on this planet rely on only about 10% of the world's surface area can produce food for us. Only about 10% of the entire surface of the planet is arable enough to produce food. And of that 10%, only 3% is stable. Only 3% of the surface of the planet has stable soils that produce the food we need. So soil stability and soil ecosystems is foundational to human society and the whole future of food on a growing planet. And Diana's work is very much involved in these kinds of issues. I'd like to add a little personal comment. This is really something Diana doesn't know, but as a professor, I teach in large auditories the size of this with lots and lots of students, and I always have them read a one page in a famous book. It's about this thick, Introduction to Biology, because it's on Diana. And anytime we talk about terrestrial ecology, I always have the students read what it takes to be a successful scientist and woman in scientist, as Diana is. So I'm extremely honored and proud to be her colleague. She sent me this little card years ago from Antarctica, which I've kept all these. It's called Changing Perceptions of Antarctic Soil Biodiversity for 21 Years. The Worm Herders, she called it. And it's stamped from I know there's a few Antarcticans in the audience. It's stamped McMurdo Station, Antarctica, January 19th, 2011. It's a keepsake. I've always kept it. So thank you for everything you've done for Antarctic science, for sustainability, and I'm so pleased to introduce the 2013 laureate, Diana Wall.
Thank you, Donald, and thanks to the Tyler Committee. I can't tell you what an honor this is. It's just extremely shocking, first of all, to be recognized in this way, and it also says a lot about what we need to be thinking about, which is soil. I thought a lot about this, as you can imagine, what kind of talk to give, and I wanted to start with one of my favorite animals. This one may not be your favorite animal, but hopefully when I finish talking, you'll love it. This is looking down the mouth of a bacterial-feeding nematode, and it reminds me a lot of a snowflake, and it could be used as a snowflake, and I think that's an appropriate way to start talking about getting you to love an almost invisible organism and climate change, and what are we going to learn from Antarctica. So I could start with Antarctica, but I want to first take a step back and give you some information on biodiversity in general in soil. And I think there's a few things that I want to put out across this whole lecture, and one of them is that the soils are alive. And we don't think about that, and we need to be thinking about that if we're going to manage them sustainably. So let's first start with just biodiversity. We all know that biodiversity provides numerous benefits to us. But I guess the question is, what do we need to know about above ground biodiversity when we see a species out in the environment? What do we need to know about them? Well, there's a lot of things that scientists can study. We need to know the current distributions of a species. Will they be able to migrate with climate change when it gets warmer or wetter or whatever? Who is the species we're looking at? You know, so who, what, when, where? Who's the species? Where is it? Why is it there? Are there limits to it? How does it respond to its environment? Does it have some kind of little mechanism that's going to allow it to survive in a hotter, warmer, or extreme environment? Or when we have these extreme pulsing events in the environment? And then, of course, who cares? What does it do in the ecosystem? So if you have some of that information, you get an idea of the response to climate change, and we can start helping to make predictions about what an ecosystem, the species in an ecosystem, will do under climate change. But what does that mean for soils? Well, soils sustain all the biodiversity we see above ground. So we ought to be thinking about soils. That's one thing that's very, very important. But is anybody thinking? Here is a quote that I really have enjoyed that says, it's from Nature Geoscience. But the scientists have yet to pay due tribute. Well, if the scientists aren't thinking about it, who's thinking about the life in soils? So we need to be thinking about it. And in fact, in international policy, there are many different areas where soils are at the center of these environmental issues. We can start with food security, which Donald has just talked about, but there's also just biodiversity. There's invasive species. What happens below ground when we have invasive species come in? What happens to nitrogen and nutrient cycling? There's also the desertification, just the changing of land to deserts as it is degraded, as we overgraze it, or we use too many plows in a particular area. There are all sorts of issues that lead to desertification. And then there's compaction, just taking heavy machinery over land and compacting it so tightly that plants and the seeds can't grow, nor can the organisms below ground move through the soil. There are other threats. Here are the threats of erosion, desertification, compaction, that have impacts not only on soil fertility and growing food, but on human health, the dust in the air, soil habitat. And you can see on the right side the sandstorms. We've heard a lot about these recently, and them blowing across the ocean, blowing forever. But it's not just somewhere else. The US has problems with this. This is Phoenix 2011 dust storm. And the question becomes, we know a lot about soils, and we know a lot about the Dust Bowl, but FDR said way back in 1935 when the dust blew into Washington, D.C., and when people were migrating from all over the Dust Bowl into California, a nation that destroys its soil destroys itself. And we need to be thinking about that. But what I want to focus on is that we need to think what helps sustain those soils. And that's kind of like the lost biodiversity, but it's so exciting. So I'm going to start showing you this part. Okay, why am I so excited about this? Well, the life in soil is so tied to those above ground species. It's not just like any old species can be there. They've kind of grown with the species. They've evolved with the species. And then we don't think often enough about wildlife and how wildlife depend on soil organisms for food, for their habitat. They may move in and out. There are all sorts of above ground bugs and insects that need the soil for their growth. We also have soil organisms that all they have to do is work day and night turning through soil, carrying pieces of organic matter or moving microbes around. So it's a lot going on to form soils. And of course, they regulate the rate of carbon stored, which brings us back to climate change. We're very interested in how much CO2, how much carbon dioxide is in the atmosphere, how much methane is in the atmosphere. So of course, we want stable soils so this can be regulated, and the regulators are the organisms. So who are these guys? These are pretty cool people. I mean, they're not people, pretty cool animals. But they might as well be, because you can tell that I'm really attached to these. But there are so many. We know E.O. Wilson studies ants. Somebody else studies termites. But these are just a small handful of the things you would see if you just went and scooped up soil. And for every one of these pictures that's up here, there are many more. Think about the millipedes, the centipedes, your isopods, that you may see the little pill bugs. Many organisms that are doing things in soil, many jobs in soils that provide us with benefits. So the question has been asked, do soils have the greatest biodiversity? And we just haven't looked at it. And this is just to give you an idea and a flavor what's there. Bacteria. People talk a lot about bacteria, and we should be talking about bacteria and archaea. 10 million species in 2 tablespoons of soil. Think about the animals in the soils also. 430 species of nematodes in a tropical forest. That's in the top inch, 3 feet square. 159 species of mites in the Kansas tallgrass prairie. I know this because one of my graduate students identified them all and said he'd never do it again. Then we have what we use now, the molecular techniques, to look at just the sequences of the various animals that are there. And in a boreal forest in Alaska, over 1,300. In the actual tundra, over 2,000 of these different sequences, these organizational taxonomic units. So there is a lot of diversity, and I just want to tell you, there's a lot of numbers of each of these species. So when you hold a handful of soil, you have got millions of species, and we don't know a whole lot of these. More than, I would say, 95% are still unknown. So what do these do? This is my favorite picture. I really like this. My graduate students don't like it very much. I don't know what it is about death in landfills, but anyway, what do the organisms do? They rip, they tear, they shred this material. They really dissolve it. They pass it through. There's a little factory going on in the soil. Everything is passed around. And they slowly take organic matter, leaves, trees, dead bodies, turn it into nutrients. And those nutrients help grow the plants so that we can have corn on our plate the next day. We can organize these into complex food webs. And so every little box you see is very much like you'd see in an ocean, only you would see the shark way over there. And that's what we've got in the last column on the right, is a lot of predators. So in this food web, there are lots of interactions among species. There's parasitism, there's symbiosis, where they're helping each other. There are tons of interactions, and this is very hard to then see what is going to happen with climate change in one species. What is going to happen with those interactions, because they're multiple. And we do know, however, that when we disturb soils, when erosion, desertification, compaction, when we plow a field, we affect the life below ground, and what we see is a much reduced food web. Each box still contains a lot of species, but it's much reduced. It's very difficult, though, to see what climate change does. So with all this complexity, I still want to turn the world upside down, and we're going to focus on soils for the rest of the lecture, which you can imagine. And I want you to see my world when I look through a microscope. The first worm I ever studied, and that's what nematodes are, so we're now focusing on nematodes, was this one on the left. And I want you to see that these nematodes, that if I put them into a glass of water and swirled the glass of water, you still wouldn't be able to see the nematodes in there. But under a microscope, you can see on the left that this guy has a little knob, like a hypodermic needle, and he pushes that into plant roots, and he can change the whole morphology of the plant root. He can change architecture of roots, and they change also the carbon flow and the water relationships of plants. So these are plant parasites. On the right is a Acrobolese. It occurs many places, but this is a bacterial feeder, and look at the ornation on it. So under a microscope, you can see a whole lot of different things, but again, they're microscopic. Again, on the far right is another bacterial feeder. But look at the difference in the two species. One looks like a corn cob. The other one looks like it's going to win the crown of something. And in actuality, they're studied for a lot of roles. Caenorhabditis elegans is studied in human health. They look just like this little worm down on the bottom, but what's so cool about them is you can see straight through them. So you know when they're eating, you know when they're starving. So there's a lot to their metabolism. So they live in films of water around soil particles, which makes it really ideal to study the effects of climate change on soils, because if there's less water, how will they react? They have to have the water to be active. They eat all sorts of things. Different species eat different things, so you have to know something about their morphology. And then, of course, though, there's the problem about how abundant are they on Earth. And they're pretty abundant. There's a lot in oceans, the blue part. We know a lot about the soil and the freshwater ones, who are green, the plant parasites, agriculture, and animal parasites, a smaller amount. And in fact, it's been said that they're the most abundant animals in soils. One person has, or a couple of people have suggested in a paper that if you took everything else off Earth and you just had a handful, 4 out of 5 animals would be nematodes. May be scary to you, but it's kind of cool. And way back in 1914, a guy studying nematodes through a microscope in a Cobb said if you swept everything away, you're going to see a thin film of nematodes. And that kind of suggests that every species would be everywhere. Well, that kind of is, again, a little hard to study climate change if there's so many species around. So I went first in my career to the hot desert, the Chihuahuan Desert in New Mexico. And what I had hypothesized there was that I would see less species because it's a desert and it's hot and it's extreme. And in between plants, there would be less species in lower numbers. And, you know, to some degree that was true. There were a lot of species I could not identify, so that wasn't helping. And if you look at this one data slide here, you'll see plant, the surface is up there where it says roots, and all the green things are roots going down about 33 feet in the soil. And then we sampled the soil, and over here on the left are the nematodes. And you can see that the nematodes were greater in abundance and decreased when you got down to about 33 meters, but in general, they just kind of follow the plant roots. And so the plants structure where the communities are. So I was already kind of in over my head with how many species are there. But there was something cool that we learned, which is what happens in their environment. This is something we need to know for climate change. I've said they're active with water, but they do the coolest thing when they start to feel that the soils are drying. They turn into Cheerios. It's a very programmed turning off of their metabolism and changing to a different biochemical pathway. And that information, one of my colleagues, John Crow, has used. Now they are thinking about doing studies using this pathway and how it changes to dry, turn off your metabolism, blow around places, and add water and they come back to life. They're thinking about looking into that for desiccated blood, dry blood that soldiers can carry. What are the mechanisms that they can use that for? But needless to say, I had too many species, too many numbers, so Antarctica. It's a cold desert. It is at the bottom of the earth. It's an unusual place for science, but let's face it, it is the continent for science. It's run by the Antarctic Treaty, for science by many nations. And a lot of people think, why would I go study a cold desert that's ice-covered? But in actuality, about 200,000 kilometers squared, about 800 square miles are just like you see, Mars. Looks like Mars. And in fact, when you land there, it looks like Mars. The first time it was discovered, so we have a lot of history, was Robert Falcon Scott. You know, a later expedition took him to the pole and he never came back. But when they went in and discovered this valley, imagine not seeing anything living then. But even when I started studying there in 1989, they said these valleys are sterile. There's nothing except in melt streams and in the ice-covered lakes. In the soil, we may see microbes, but there's no animals dying. So we took a gamble, my colleague Ross Virginia and I, and said, I bet there's nematodes there. Nematodes are in a thin film around the Earth. They got to be there. So we took off and we went to this analog of Mars, to this extreme desert ecosystem. And I just wanted to give you some numbers about how cold it is. This may not look cold to you all, but it's cold. 5 months of dark, 21 days above freezing. Very little water and very poor carbon soils, but I should say it is a desert analog and that the pH is alkaline. This is one reason we chose it. It's very similar. Not only is the pH alkaline, high nitrates in the soil, it is very grainy, as you will see here. Lots of soil, and the landscape is predominantly soil. And then right there is a person to give you a perspective. And of course, the question here is if there are no plants, if there's no bees, if there's nothing moving around, there's no sound except when the wind blows tremendously, how in the world are we going to go find what's living in these soils and then be able to do what I was hoping we could do, which is to just find a few soils with a few species, and I was hopeful we'd find more than one, and then soils without, and we'd be able to say, well, why aren't there nematodes or any animals, I didn't care what we found, in these soils with nothing? What are the limits there? What makes them such an uninhabitable place versus soils where we would find it, find the worms or whatever we were looking for? So we sampled a lot. We sampled everywhere. We walked that landscape and we looked at all sorts of habitats. And one of the great things about deserts is their soils are very heterogeneous. And in the dry valleys, they're even more so. You can see different soils just like they were left thousands of years ago. And of course, I wouldn't be standing here if we couldn't find some fantastic nematodes. I would be working somewhere else probably. But this is what we found. Nematodes, we've now got about 6 species of nematodes. This one in the center, big bacterial feeder. Over there is a microarthropod. There's about 3 species around melt streams. This one that's kind of glowing at the bottom is a tardigrade, a little water bear. There's about 3 species of those. Little rotifers. Those guys are all in water. But what I really want to impress on you is this is it. We were really lucky. We found a place where there's one species here and one species there, and we could start to test what do they do and how will they respond to climate change. E.O. Wilson has called these guys the invertebrates, our equivalent of lions and elephants and tigers. But I mean, just think about this. There's nothing higher there. There's not ants, there's not termites, there's not millipedes, centipedes, all that stuff. There's no plants, it's cool. So we have a place that looks on the surface like autoclaved glassware, but when you get your microscope out, it's amazing what you see. Are all species everywhere? No. Scott Nema, named for Robert Falcon Scott, has the highest abundance, and it is the only invertebrate out there in those dry soils. So each species, just like a lion, an elephant, and a tiger, has a very different habitat defined by that soil and the characteristics of soil. Physical factors, not plants, structure communities there. This predator of bacteria, Scott Nema, stops growing and being active when the soils get very saline. When they get too wet, it doesn't like it. When there is too much carbon in the soils and they're wet, it doesn't like it. So its range we can look at and answer the who, what, where question. We've got a lot of information. Eudora Limas, this is another species. I think it's kind of ugly, but it's okay. It's restricted to a wet soil habitat. It's only found along wet areas, very restricted. Feeds on algae. You can see the chloroplast there. But the other thing that we were able to do, and when I say we, I mean a collaboration of a lot of students, a lot of postdocs over the years, and a lot of collaborators from very many different disciplines. I want you to watch this. This is coming out of anhidrosis. The mechanism was similar in the dry valleys as it was in the hot desert. When it desiccates, it goes in to this anhidrosis, loses 99% of its water. You can drop it in liquid nitrogen, you add water and it comes back. And this is what it does for winter. But it also has a suite of other genes that we have studied that we can see now what upregulates when it's just a cloud passing over, which antifreeze genes are turned on. It's a very amazing animal, but what's even more fun is we can now predict, based on all the data we've taken on soil moisture, and where you see the little red dots, that's active worms at a certain soil moisture, and we know exactly when it is, and when they get to be black dots, they're in anhidrosis. So we know at what moisture level, what their physiological mechanism will turn off or turn on with climate change. We can suggest that. So let's look at some of the climate change. As many of you may know, it's been warming on the Antarctic Peninsula, but where we study is down here on the Ross Ice Shelf in the Transantarctic Mountain Range. And over a period of years, it has been cooling there. But nevertheless, we've seen some warming pulse events. So along in 1995, we started studying what would happen with warming. We put these little greenhouses down, these little warming chambers that are affectionately called worm farm, because we're always going out to sample and see what's happening there. And we warmed the soil about 2 degrees, and the soil dried out even though it was cooling. We've kept those. We have these scattered out throughout the valley. We're able to have treatments with water, without water, with temperature, without temperature, the combinations. We've added carbon to some of the soils, so they have a substrate for a food web. We've also just put up simple snow fences, and this increases the duration of water. It takes it a lot longer to melt, and so we have more water going into the soil. And then we can see what is happening to the invertebrates there. Well, make a long story short, it's declined over a period of years since 1983. Scott Nema has declined considerably with long-term cooling. The little red dot is just for me to remind you that we do have, even with cooling, some extreme pulse events. And so we've also been able to study what happens if you warm it too much, and Scott Nema declines. So when the soil's dry, Scott Nema declines, and when the soils get wet, Eudora Limas increases, because Eudora Limas is the wet nematode. So we've got one declining with warming and cooling, another one, when it's warm and wet, it will increase. This has happened across the landscape. This may not look phenomenal to you, but it was pretty startling to fly out and see in the middle of an experiment that I had that had no record of no flooding across the soils in Antarctica. Here on the right-hand side, you see a river flowing across the landscape, and we were able to study that. And we have seen that the species composition has been changed. So does it matter if this declines? Well, we've done a few experiments. This is one where we feed the soil a C13 cocktail. So it's a tracer. We're going to follow it through. The microbes take that up, and then the nematode eats the microbes. And then the nematode has the signal of that cocktail. And what we found was about a 7% carbon turnover due to one nematode. What does that mean? The carbon stability in that soil, how much is flux to the atmosphere and stays in the soil is due to one nematode species. So, does it matter? Well, that's 65% decline from 1993 to 2005. Potentially a 30% loss in soil carbon cycling. Reduction at the top of the food web. The predator of bacteria is gone. So does that mean there's going to be more bacteria? And what are they doing? Or they're going to stabilize? So the question becomes, should we worry? Should we not worry? I mean, let's face it, this is our polar bear for the dry valleys right now. And there are a lot of other reasons to be concerned about conservation of Antarctica itself. But what lessons have we learned? One, keep looking, discoveries beneath us. Species aren't cosmopolitan in soil. They have their own area. They have their own range. They don't, even if they're blown to an unsuitable habitat, they won't live if it's too saline for Scott Nema. So soil habitats are very important to species of soil animals. There are limits to their adaptation. If climate change, when climate change changes the dry valleys in many years, there may be limits to whether or not they'll survive. Scott Nema will. And we found that very much like the dry valleys, where a small change in temperature can be magnified, a small change in a single species can have a magnified impact on the environment. So climate change reduces the top species, the predator, and alters the soil carbon cycling. So now I'd just like to take these lessons and show that I think they have a pretty important effect on the science that we're starting to do other places. And so do these lessons from this polar environment apply anywhere else? Some of the work we're doing is to look at bacteria across each biome, or as many biomes as we can. And all I want you to do is to look at one side of the line. There are clusters of red and there's a cluster of brown at the top where things that are closer together are more closely related. On the other side are a lot of different biomes: tropical forest, coniferous forest, boreal forest, Arctic tundra is on the right-hand side. And what we've found is that deserts are different. The bacteria in deserts, cold deserts, is different from hot deserts, the ones at the top, the triangles. And they're very different. Then we can look at the metagenome and say, what is the difference with these different genes? The ones on the left are upregulating more on osmolarity, trying to survive in a desiccated environment. They're doing more with dormancy. There's a prevalence of those genes that have to do with dormancy or upregulation of that. And on the right-hand side, where we have forest and tundra and hard material to degrade, the genes that are upregulated, the metagenomes, are very much more prevalent for carbon, nitrogen, and sulfur cycling. The second example is this Africa example. And we're really very excited about this. I mean, one of the things is generally there's somebody who studies bacteria, there's somebody who studies caterpillars, somebody who studies pill bugs, somebody who studies nematodes. Here we are trying to take a soil sample and tell you everything that's in it. And we think we are on the forefront of doing that. What we've got along the bottom here is just bacteria. So very many different bacteria in the sub-Saharan soils. We also are able to look at all the fungi and animals. And you can see that curve just keeps going up. So as we've gone from 0 samples out to 120 samples, the more we sample, the more different types of sequences we find, high throughput sequencing, the more organisms we're starting to see, most of whom are not known to science. But we don't want to just find things, we don't want to just monitor things. What we've been working with is with our colleagues, and I've got to say this is with the African Soils Information Service, from a Gates grant to them. And we work with them on the same samples they do and where we want to do, and this is just an example. This one's going to be a little, I've got to explain this one to you. 2 sites, 2 different sites in Tanzania that we took the soils, the blue and the orange sites. And what we've done is look at all the soil properties. That's not us, that's the African Soils Information Service, but we exchange our data with them. And so we can see that there's some differences in the soils in these 2 places, and some things that are in the soil. So their habitats could be different. Then what we do is look on the far right top, and you can see those dots again are separated. There's orange and blue dots, and those are two different sites saying that they have kind of their own total soil biodiversity. We then have the maps that are predicting temperature, moisture with climate change. What's going to happen in Africa across the sub-Saharan? And we put it together and say, okay, do we see anything we recognize in there that are fungal pathogens, human pathogens, animal pathogens? We could not have done that 10 years ago. We weren't as savvy about thinking our soil habitat's different for different soil animals. Now we are, and now we're approaching the forefront where we can reach into a soil and see everything that's there and use it for not only pathogen detection. On the left is the fungal blastocystis, and on the right is a nematode plant pathogen. That doesn't mean that they're going to be active and out there being a pathogen. There are going to be other conditions, but it's presence, absence, and which site has more. And then we're also looking to try to bring it home so people are interested in soil biodiversity. And this is a project that a number of us are doing. We went in one day last summer in July and sampled Central Park, and it was hot. And it was 680 samples. I'm sorry you can't see the little red dots very much, but 680 plus samples all over that place. And by nighttime, we knew where every sample was. And of course, my colleagues are looking at the soil characteristics of each habitat across Central Park. And we have gotten Illumina to help us with the sequencing so that we're doing bacteria, fungi, archaea, and all the soil animals for that. And the data isn't coming out yet. I mean, it's coming, but it's a lot to put together right now. And so I couldn't get it for this talk. But it is something to take this soil biodiversity and bring it back home and say to people, we know growers and farmers are extremely appreciative of their soils, and they know a lot about what's in them and what to put on them. But let's take it to the other people who haven't been excited about the discoveries you can make. The new scientists are thinking, what frontiers do they want to go to? And I'd also kind of like to go back and think about that Nature comment that I quoted, Nature Geoscience comment I quoted, where it said that scientists should pay soils their due. And I'm got to say in the last 5 years, things have changed a lot because a number of my colleagues internationally came to me a number of years ago, 2 years, and said, Diana, we've got enough information on soil biodiversity in different biomes to start thinking about how we're going to manage that soil food web. How can we manage that for better agriculture? How can we manage that in forest? How can we manage that soil to help form soils, restore soils, prevent pathogens? Let's start thinking on a big scale. And so we recently had a meeting in Fort Collins, and they came from international all over the place. And the EU has taken on the charge of saying that they will pay for an atlas of soil biodiversity globally. So we are going to attempt that in 2 years, and we hope eventually that there will be an assessment of what we know now. We need to know it now, as we've seen with climate change. If we're interested above ground in what is the distribution of the native species now, what is its physiology and how it's going to respond to the environment, we need to know for a bigger area, and we think the scientists are ready to do that. So I think those are kind of 3 to 4 things that I think take these applications from Antarctica and move them into other areas. So it does matter if you do research in a frozen desert. To me, it has been the comparison of what we've learned from 1 and 2 and 3 species across that dry valley, and how we can then translate that into other systems, compare it to more biodiversity, and think about the benefits that soils provide. So I'll go back to this, that the life in soils sustains the biodiversity we see, but everything we see is tightly connected to the soil and the life in soil. We cannot talk about soil like it's Mars. And I think the Antarctic lessons that biodiversity and soil must not be ignored in global environmental issues. They not only help to clean the water and the air, but they do a lot of things that we haven't explored. And if we want to prevent these kind of environmental disasters, we have to think more carefully about who's there and how we can manage this. So with that, I'd like to thank a whole lot of people. I won't take too many slides to do that, but the worm herder team over about 24 years or so. They've been fantastic. It's been a very interesting bunch. And the McMurdo Dry Valley LTRs, the PIs from there, the principal investigators from there, are all flying in today to be here for tonight. And I think one of the things about that is that working with this bunch, when you work interdisciplinarily, like we do on the ice, to work with a geophysicist who doesn't know what a nematode is, let alone how to pronounce biology, we had a lot of learning to do because I didn't know much in the other direction either. And we have glaciologists, and we all sit down and say, what is happening to this ecosystem with warming? Why are the lake levels rising? What is happening with the temperatures? So the intellectual exchange is much faster, I think, when you have a group of collaborators all thinking about an ecosystem. And so here are just a few of my colleagues, lots of red coats, but particularly to remind me that there's an international contingent to working in Antarctica. I mentioned that early, that it's many nations. The Antarctic Treaty, every nation has a place, but we on the ground level and also at top levels exchange a lot of information back and forth about what's happening, not only in the dry valleys, but in other areas of Antarctica. I want to also be reminded to thank the support that we get. We could not do it if there were not people who want to go to Antarctica and support the science that is done there. And they often don't get recognized, but I just want to be sure that sometimes you never know. One of you may be down there as a plumber or sweeping the floor because you just wanted to go to Antarctica, and their support of science is pretty incredible. So thank you very much.
And he noted the example of the Antarctic continent, notwithstanding the West Antarctic Peninsula, but the actual region where you were studying as being one of those distinctively cooling areas. And he used that in part to deny that there was any kind of generalized warming trend. And I'd just love to hear a little bit about your response to something like that, because I was completely incapable of responding to who is wrong.
Yeah, well, I've got to just put aside also that when we published a paper in Nature about, what was the response of cooling, the whole group of us, we got off the ice and went to the bookstore in Christchurch. The first thing we saw was Michael Crichton's book, who was denying climate change and using our paper to say that that's why. And I think that, and I'd love to bring Richard Alley into the conversation here. But my latest recollection on this is that we're seeing extreme warming events right now, and that the long-term projection is still for warming. Richard, would you mind saying anything? I want to take advantage of our colleagues here who are knowledgeable about this.
So West Antarctica is now warming very rapidly, as well as the peninsula. Probably the cooling that you saw in the dry valleys is some combination of natural variability and the ozone hole. And the ozone hole really does change the circulation a little bit. There's also some things propagating
You got it from the real authority. Yes.
Professor, to me, you bring new meaning to the word global warming. Global warming.
I wish I'd thought of that.
Two quick questions. One, if you had the president's ear, what policies besides reducing the underlying cause, reduce petroleum use and try to reduce the impact of climate change, or the cause of climate change, what policy could be brought forth to protect the life in the soil as we know it? Is there one other, a handful of other human factors involved, the use of herbicides and pesticides and those things? What would be the policy you would want the president to institute that could protect the life of the soil?
It's very interesting that you asked that, because the EU has just gone through, about 3 years ago, they finished and put out a report and have tried to get soils to be listed just like the Clean Air Act is here. In other words, they are moving fast. They've got one nation that's holding out, but they are moving fast to say soils are a non-renewable resource and that we have to pay attention to policies that we already have ongoing that are helping to destroy soils. For example, one of their, and I'll get to your answer, but what I think, but it's written out, you can see it, they've got step by step by step why, and why they want a Clean Soil Act, or whatever they wanted to call it, a Soil Protection Act. And these things include sealing over with concrete our most fertile soils, this small percentage that you're talking about, not paying attention where we're building our cities. Just, we could just build anywhere. It's not just because of water. We need the water, but to grow crops and to use soils, we've got to have the combination. We've got to think about it in a sustainable picture. So I could give you the reference to that, to look at that. And my push is to say, of course, there's life in soil that we need to discover, and it's going to provide a lot of benefits that we don't even know about. We've got to use the knowledge we have to restore some of those soils. And we also, in agriculture, are moving towards, there's huge no-till soils, but there's also an emphasis on getting rid of chemicals and fertilizers and the nitrogen that goes into that and also pollutes water miles and miles away. So I think it's a combination of things that we're already doing, but with just a focus on soils are like air and water. We may not be able to breathe them and eat soils, but we don't get food on our table if we don't have soils.
If I could have a follow-up question.
Yeah.
Your last comment related to protecting the soils, and is there any work on maybe a business case where there's the soil augmentation or soil enhancement, having samples that farmers could use with the nematode population of a certain soil to add to the soil to make it healthier?
I mean, they've been doing a lot of things, and Dan Bush and other people here in the audience could help me out on this, but they've been doing a lot of things in the past 20 years to change this. And it's not just adding one, it's trying to build up, say, with a certain amount of tillage for a particular crop, how you would get that organic matter to build more complex food webs. And even sometimes with tillage, you're going to have to apply a herbicide. So it depends. I mean, I hate to say it depends on this, it depends on that, but in actuality, soils are different, so we do need to depend a little bit on this and a little bit on that. You can't just take one nematode dried up species and put it across a landscape. You can go to the garden store now and buy dried up nematodes and sprinkle them on your yard, and they're the ones that parasitize insects. And so it's kind of a clean, organic way to, get rid of bugs in your soil or something. But I think that, I guess I really want to emphasize it, that where I would see protection is that when we're going to say we're conserving a national park or a particular area, that doesn't necessarily mean that the biodiversity below ground is preserved. We do not see, like we see for the tropics, more species in the tropics. We see that Kansas prairie has many more than what we found when we went to the Amazon in Peru and looked there in this global study. And people are looking at the same thing for bacteria. I mean, we did it just for soil animals. So I think there's a misconception that just because we're saving a particular area, we're going to be saving those organisms below ground. And I think we now have to rethink that. Jonathan.
Yeah, Diana, I'm interested in your mapping of the soils, and you showed Africa and how the soils are differentiating between parasites and fungi, and it depends on the moisture. And I'm very interested, of course, in the effects of climate change on human health, and one of the biggest issues is food and malnutrition. And I'm wondering if you know if the models for malnutrition and crop yields adequately adjust for how the soil microbes may change. Now, of course, the one thing you mentioned is fungi and parasites, but do the crop models for climate change scenarios actually take into account changes in soils that you're talking about?
I know that if you go to, well, I can give you the website. If you go to a website, they do predictions of soil moisture in Africa. You know, they kind of, it's like, what is the soil moisture today? Just like they do here for crops or something like that. So here's what it is. And you can go to this one website, I think it's out of Wageningen in Holland, and you can see the projections of what soil moisture will be based on some year average they're adding to. But I don't know at what level that is. I don't know if it's just, broad swath across, or if it's by biome. I don't know the details on that. And then I do not know how it ties to crop models, no. But I don't know if Dan knows. Dan, do you know?
I don't think the soil's a black box for most crop models that I'm aware of.
So, I mean, I'm just thinking as far as direction, it's all about soil moisture and crops, but then however that change in biodiversity of the soils, that could be an actual bigger effect from the climate, possibly.
Possibly, yeah. I mean, it's something that we're just, I mean, this is stuff that we're at the forefront on, doing total soil biodiversity. I've always wanted to be just kind of a bumblebee and fly over the soils and not have to sample it, and then for every sample, it's like 6 hours on a microscope, you know? Wouldn't it be nice to be a bumblebee and say, oh, high diversity here, high diversity there, and then try to see, okay, what is the pathogen load, that kind of thing. And it's just not there yet, but I think we're on the forefront with what we're doing now. Wendy.
I'm interested to know whether any of this science that you're doing is filtering into any of the big NGOs, the Nature Conservancy or other big, or in our national parks. I mean, is it becoming part of the intellectual framework as people look at that?
I think it is, because I know that there's a woman in LA, I think it's the Nature Conservancy, who writes beautiful prose on, why aren't we paying more attention to soil biodiversity? And certainly I've been contacted by people who are interested in endangered species, who that particular animal uses soil as a habitat, and the habitat's being destroyed, very much like birds who use soil as nest. And so the linkage is, in my ears, it's more, but I don't have any data to show. I can tell you that in the scientific world, the numbers of papers on soil biodiversity in the last, I would say, 10 years have just gone skyrocket. So the tight coupling of plants and what's below ground, because people are thinking, okay, well, if the seeds of a plant start to move with climate change, they don't think, well, is everything down here going to move, or does it matter? And there have been some studies to show that a home field advantage, having these things tightly linked, makes the rates of decay faster than if you move over here to another habitat with your seed and try to grow your plant and the microbes aren't there. So I can't answer it totally about NGOs. Yes?
So one of the important parts of Al Gore's message in Inconvenient Truth was finishing the movie with simple things that anyone could do that would have some sort of positive impact on global warming, climate change. What are some simple things that people can do that would improve the fate of global soils?
Well, I think this is my personal opinion, okay? This is not backed up by a ton of data. It's my personal opinion. My personal opinion is think before you pour anything in the soil. You know, it's just like a regular landfill, and where's it going? Think before you over-fertilize your front yard. And if you see somebody in a garden store saying, wait, you know, what are you thinking about putting on this soil? We don't want the nitrate to end up somewhere else, but it also doesn't help the organisms in the soil. I think there are even more. If you look at if you're influencing a board or a group of people, let's talk about why not put some experiments out just for people. Let's say, I'm going to dry my yard so much, and let's see if it rejuvenates, and let's not. And let's see the difference in comparison, and can we live with what that more natural, hopefully natural system is right there. And so we're not constantly trying to go for the green monoculture. And if I had my way, I guess I would, this is going to be awful, but I think the new monocultures are golf courses. And I think, you know, we really work to keep them all great. Let's learn to play golf with a little hardship, and let's let that monoculture go some and not have to fertilize it and pesticide it and whatever. And I guess, you know, that's not going to happen. But I'm just thinking, no monocultures. Let's go for more of the diversity above ground. And of course, you know, using compost to a certain level. There's all sorts of things you can do in your own yard. Mm-hmm.
So you mentioned Mars a few times, and as somebody who has studied the closest, essentially, ecosystem on Earth, the closest to Mars, I'd love to hear your thoughts on the potential prospects of finding evidence of life or discovery up there now, taking soil samples, and what you think that might look like if in fact they do. Evidence of a form of life.
Well, I guess, I mean, I don't really know where they are on that, although we've got a lot of NASA colleagues that come to the ice, and we talk with them. If somebody's here who can answer that more, I mean, my personal impression is that I want them to keep looking, because just like in the dry valleys, when I went there in '89, we knew that there were things along the melt streams, but we certainly didn't know about that dry, barren environment, that we would find anything there. In fact, we laugh about it now and say we thought we had a one-year grant because we knew we were coming home. There wasn't anything there. But the NASA guys, really incredible about taking some of our data and saying, okay, now we want to look within rocks. Now we want to look within places where you wouldn't expect, where you might see the signature. They've been seeing the pathway of Mars. I think they're going to see where there was life. I don't know if they're going to find anything beneath it. It's kind of, to me, it's very, very cool to find out that they can bring nematodes from 2 kilometers, you know, that gold mine type story that was where they took I think it was 2 kilometer core down from a gold mine that they were looking at and brought it back supposedly sterile, and there were nematodes in that. So I can't equate it to Mars, of course, but that would be my hope.